All right, there we go. All right, welcome back to Invertebrate Zoology. And I'll start presenting there. That looks like the one. We'll finish up with the protists and start talking about what makes actual animals, and hopefully get a pretty good ways into the sponges, the simplest of the animal phyla, before we're done. So we've actually finally gotten out of the eukaryote weeds into the kingdom of the Animalia by the time we're done. So just as a review, something you've seen before, we have this scheme of 5 supergroups. We've talked about amoebozoa. We finished off SAR on Wednesday. All of the critters that we talked about on Wednesday are in the alveolata subgroup of SAR. The last group I'm going to talk about is Excavata, which is important in, among other things, in medicine and veterinary medicine. There are plenty of free-living excavates, but there's also lots of parasites as well, and symbionts. Archaeplastida and Opisthoconta are the others, and we'll finish with those today. Excavates mostly bear elongated flagella, and I mentioned this before, but just in case you didn't catch it, Lynn Margulis pointed out that eukaryotic flagella and cilia have basically the same structure, but they're both very different from bacterial flagella, and she coined the word undulopodia to mean eukaryotic cilia or flagella, there being no real difference between the two except length. This is never particularly caught on outside of Margulis's circle. In some books, you might encounter undulopodia to mean specifically eukaryotic flagella. I don't personally, maybe it should bother me more than it does. I don't personally care. If you want to call these flagella, I will not object. But do know that the structure of eukaryotic flagella is not at all the same as what bacteria have. Eukaryotic flagella wave back and forth. Bacterial flagella actually turn like a screw. So different mechanisms there. Anyway, most excavates have flagella. Many have a specialized feeding groove, although not all do, but that's where the word excavata comes from. They'll have a feeding pit excavated, as it were, out of the cell, although you don't see that universally. There's many that don't have mitochondria that function now, but they do retain DNA sequences that were transferred from ancient mitochondria. So they used to have mitochondria. In some cases, the mitochondria have been modified into new types of organelles, like hydrogenosomes, which ferment pyruvate into acetate and generate some ATP and also generate molecular hydrogen, generate hydrogen gas. There's plenty that are perfectly good free-living organisms, but as I've mentioned, there's plenty of parasites, pathogens, and symbionts in the excavata. So there's a human symbiont. This is Trichomonas vaginalis. This is a human commensal that in low numbers doesn't seem to cause problems. In large numbers, it can cause a vaginal infection that's not particularly dangerous, but from what I've seen is kind of miserable. I used to see these guys on a fairly regular basis because I went to grad school in the People's Republic of Berkeley, California, and a couple of months after I arrived, there were riots breaking out because Berkeley has them every so often just to keep in practice. This was actually over a local issue, although there were riots again in the aftermath of the Rodney King verdict. I think we had 4 major riots during the time I was there. And I actually went down, my first riot. I'm kind of excited. And I brought down some medical supplies, and I ended up meeting people working with the Berkeley Free Clinic. And I ended up as a lab tech working there as a volunteer for several years. So, yeah, my main clinical experience, I got my start as a riot medic. Although the Berkeley Free Clinic saw a lot of homeless people, also a lot of students, and just, working class and unemployed people. And we treated a heck of a lot of sexually transmitted diseases. I've seen humongous amounts of gonorrhea. And we also looked at women's health problems. So I've seen a lot of vaginal swabs and smears. And on any given night, I had pretty good chances of seeing live Trichomonas vaginalis in a human vaginal sample. It's just one of those things. And this is pretty well what they look like. They could look a lot more round, or they could look kind of pear-shaped. And you have 4 flagella. 3 of them extend out freely, and then one of them actually bends back, and it's attached to the cell body by a little extension of the cell membrane. The whole thing makes up a sort of fin. And you could actually see this little fin kind of sine waving back and forth on the surface of the live Trichomonas. It was very distinctive. There's really nothing else that we'd see that looked like that. There's a structure made of proteins that forms a rod that runs through the cell. It's called the axostyle. And in these, you can actually see it protruding out of the bottom. That red thing sticking out of one of the cells, that kind of at the center right, is the axostyle. Under light microscopy, it's not quite as easy to see. But I did see that in my live Trichomonas as well. This is a close relative. These have been stained and shown under light microscopy. You can see they've all got this kind of darker colored ruffled spot. That's that fin. That's that flagellum that's attached to the rest of the cell body by this web of plasma membrane instead of being able to stick out and beat freely. You can see the free flagella here pretty well as well. Trichomonas fetalis is a cattle parasite, and it's one of the leading causes of spontaneous abortion because it can move from the cow's vagina into the uterus and trigger abortion that way. For the record, to the best of my knowledge, the human Trichomonas vaginalis can't do that. But it can here in the cow parasite. And it can even be transmitted in semen samples because it can survive freezing. An awful lot of cattle these days are not impregnated in the good old-fashioned way of just keeping a bull in the pasture, which is the way they used to do it. They are artificially inseminated, and you can actually buy semen samples from champion bulls. There's something of a market in it, but this can actually be transmitted in frozen semen. So, strange new horizons in sexually transmitted disease, I suppose. I think Trick vaginalis can be sexually transmitted as well. It doesn't live very long in men, but it could live just long enough that it could be transmitted that way. I'm not entirely sure how common that is, though. Anyway. Other members of that group, the Parabasalia, are termite gut symbionts. We saw what I think are some Trichomonas symbionts in that termite gut that we looked at yesterday. We did see Trichonympha. These are 3. One of them, the one at the lower right, you're actually looking straight down on that nose cone type structure called the rostrum. And then the other 2, you're seeing more from the side. You can see Trichonympha has lots and lots of flagella. kind of a hippie protist there. And in one of them, the one on the left and lower down, the nucleus is kind of in the middle of the cell, and back of the nucleus, posterior to it, you can see wood particles that have been taken up by the cell, where they're getting broken down by symbiotic bacteria inside the trichonympha, inside the termite. So we've got a nice case of Russian doll symbiosis here. There are lots of excavate symbionts in termite guts, and exactly what you've got depends on where you are. I said in lab yesterday, we found that odd kind of elongated and somewhat spirally looking gut symbiont. And I said that when I saw these in lab back in California, we called these strablomastics. That's apparently something you get in Western termites. In Eastern termites, it's likelier to be something called Piersonympha. But there's other things it could be. And different termite species have different sets of symbionts. So I'm really not sure what that was. But this is one thing you could see. Divescavina has flagella, looks a little bit like a trichomonas, but the flagella don't actually beat. What enables Divescavina to swim is thousands of symbiotic bacteria that are stuck on its surface. So it's a little bit like powering an airplane by gluing a million houseflies to it. I'm not entirely sure that would work in real life, but that's what Divescavina does. You might recall that I mentioned a while back that Lynn Margulis came to think that eukaryotic flagella or Ungulipodia or whatever were descended from symbionts, just like mitochondria and chloroplasts were. Well, last I looked, she probably wasn't right, but this is where she got the idea from. It's not as crazy as it might sound, because there are, in fact, a number of protists, most of them termite gut protists, that swim because they've got swimming bacteria forming a symbiosis with them, stuck on their outside and beating back and forth to propel them through the termite gut. So that idea doesn't have a lot of support right now, but it wasn't as far-fetched as it might seem. There are protists that use symbiotic bacteria to swim. This is actually another one that does that trick, Caducea versatilis. At the top left, you're seeing it under phase contrast light microscopy. Over at the top right, you can see it stained fluorescently, and every one of those little green spots that you're seeing in picture B on the top right is a bacterial symbiont that's stuck onto Caducea versatilis that enables it to swim. The cool thing about it is that as it swims, its nose cone actually rotates clockwise. In fact, Caducea used to be referred to affectionately as rubberneckia because its anterior end is constantly spinning, kind of like that scene in The Exorcist, I guess. And the reason this might actually matter to anybody who's not obsessed with termite guts is that when this was discovered in 1976, this was a key piece of direct evidence for the fluid mosaic model. That's the model that you probably learned for cell membranes, right? You have this lipid bilayer membrane with phospholipids with a hydrophilic head and a hydrophobic tail, forming this double membrane with the heads all facing out and the tails all facing in, right? And embedded in this film of phospholipids, you've got various proteins and glycoproteins and things like that kind of floating around in this two-dimensional film of fluid. This was a key piece of evidence for that model, because if the cell membrane is, a solid membrane like a rubber balloon, you couldn't have part of a cell constantly rotating around another part, right? Just like you can't take a balloon and twist it without causing it to pinch off or possibly to pop very quickly. This is evidence that cells don't have, solid but very thin and flexible membranes. What they've got is this lipid bilayer, an extremely thin and physically constrained film of liquid, more like a soap bubble than like a balloon. So this is why it helps to know the weird and the obscure and the strange and the, oh my God, when are we going to need to know this? Every so often, it's the weird and the strange and the obscure that are the source of the breakthroughs that are uniquely able to provide information that you didn't have before. So there. Sorry, that's kind of a popular soapbox for me to step on. I'll try not to do it too often, but it's true. Okay, so Parabassalia is the group of excavates that includes most of the termite symbionts. Another group of excavates is the Fornicata, hardy har har, and the most notorious of the Fornicata is Giardia, Giardia lamblia. It's currently considered one species, although it's suspected that there's probably a lot more. It's the cause of a rather nasty intestinal infection called hiker's diarrhea. And there's a light micrograph of one over there at the left, and it's actually a paired cell. There's a septum dividing the cell in two, and a pair of nuclei, and a little, sometimes called a clawhammer body in the middle, meaning that when you see these things, they almost look like they're tiny smiley faces. That's one of the things that microscopists look for. Somebody in parasitology did a poster. They called it, the parasite that smiles back at you. So that's pretty distinctive in fecal samples. Over there on the right, you've got a scanning electron micrograph of one, and you can see flagella coming out, and on the underside, there's this broad, flattened area that acts as a little attachment disc, almost like a suction cup, that causes these cells to attach to your intestinal lining. These are passed in, you can find living ones in fresh feces, but they also form cysts, resting stages with thick walls. And when those wash into water and a host drinks the water, they get, the cysts pop out into trophozoites, the active forms, and reproduce and fill up your intestine. And they look like this. That's a piece of intestinal lining, and every one of those things that looks like little caps is a separate Giardia cell. Severely infected person may pass 14 billion Giardia in every stool. The primary effect of this is bloating, cramps, and a rather miserable type of diarrhea that I usually described with words like foamy. You do not want to get this if you can possibly avoid it. I don't think it's fatal, but it's very unpleasant. Giardia can infect, I think, at least 40 known species of mammal. And it's called hiker's diarrhea because hikers tend to get it if they drink water from, pristine mountain streams. If you're hiking in the Ozarks and you come across a babbling brook or a little river or something like that, you might be tempted to scoop up and drink its crystal clear, pure waters coming forth from nature, And that would be a big mistake. Because even clear-looking water can carry giardia cysts, because you know what bears do in the woods. Animals defecate, and sometimes they defecate in water, or they defecate and feces wash into water, and giardia cysts can end up contaminating what looks like clear water. Hence the name hiker's diarrhea. You'll also hear it called beaver fever, because streams where beavers have built dams tend to have a high risk of this because it can colonize beaver guts as well as human guts. So when you're hiking, boil your water or use the filter or use the tablets. Don't drink unfiltered or untreated water if you're out in the woods. I mean, unless you're desperate. But yeah, if you're going to go hiking, be prepared. So there. Third group of excavates, euglenids. They're excavates whose ancestors picked up chloroplasts from a green alga. Genus Euglena is actually pretty common in, I've seen it in pond water around here. I don't know if we will, but it's grass green and usually has a shape ranging from, pointy and elongated to more teardrop shaped. And it's photosynthetic, but it's perfectly capable of feeding as well. It's got one long flagellum and one very short one. And strips of protein form a support just underneath the cell membrane. It's called a pellicle. And it means that euglenas maintain their shape pretty well. They can flex a bit, but they're comparatively rigid because they've got these protein strips. On the right, you've got a non-photosynthetic euglenid. You can see the flagellum very clearly here. That particular one is called Paranema. And it's also pretty common in freshwater around here. Closest relatives to the euglenids are a group called the kinetoplastids, and I'll explain where that group comes from in a minute. There are plenty of free-living kinetoplastids. They're small, freshwater, little flagellates that just kind of, swim around and don't really do any harm or anything like that. But the kinetoplastids also include some parasites. If in lab 2 if you happen to catch flies, this doesn't work every time, but if you catch a fair number of them, it might be worth it to try to pull out their front guts, their foreguts, by very gently applying traction to the head and making a squish mount of that, because there are kinetoplastids in the genus Herpetomonas that infect flies, and you don't get them in every fly, but sometimes you can catch them live. And that's cool because they're close relatives of Trypanosoma, which is over at the left. That's a blood smear, and you can see round blood cells that are stained red. Those nearly circular red dots are blood cells, but in and among them, you can see these kind of bright fuchsia cells that are shaped kind of like S's or sigmas, very curvy. That is genus Trypanosoma. There's a South American species of that causes a chronic illness called Chagas disease. And there's one in Africa that causes African sleeping sickness, which if untreated, humans can get this. And if it's untreated, it ends in lethargy, dizziness, and eventually a coma, and eventually death, hence the name sleeping sickness, because you slowly drift off into a coma if you have this. On the right, leishmania, close relative. In places like the Middle East and Southeast Asia, Leishmania can cause very severe internal infection, internal disease called kala azar, which can really do a number on your spleen. And in fact, it can be fatal. In South America, other species of Leishmania can infect the skin and cause very severe ulcers. I decided I'd spare you the picture of what these look like, but it's kind of gross. Both of these, by the way, are transmitted by the bite of flies. You get trypanosoma in Africa from the bite of a fly species called the tsetse fly, Tsetse. In South America, you get Chagas disease from the bite of a blood-sucking bug called the benchuca. And you get leishmania from the bite of sandflies. So all of these are insect transmitted. Herpetomonas is the one you can find in houseflies around here that I just mentioned. And that, as far as I know, does not infect any fly that bites. So humans can't actually get it. Where the word kinetoplast comes from can be seen here. You're looking here again, this is more trypanosoma swimming around among stained human blood cells. Those round things with the light center are human red blood cells. And then the crescent-shaped things are, the trypanosoma. The nucleus is a fairly large spot, roughly midway down the cell. If you look, there's 3 of them that are closest to the very center of that slide, and they've all got prominent nuclei more or less in the center. But then if you look at that flagellum, in trypanosoma, although not in others, the flagellum is also attached to the cell body by a fold of cell membrane, giving you that fin. As far as I know, this should be convergent with what we saw in Trichomonas, but it's the same structural idea that the flagellum is held in place by this webbing forming that fin. And in some of these, you can see the fin kind of S-curving back and forth pretty clearly. And right at the root of the flagellum, there's a second dark body, smaller than the nucleus, and that's called the kinetoplast. And the kinetoplast is cool because it's pretty much the only place that you can spot mitochondrial DNA. I mentioned a ways back that we think mitochondria are descended from symbionts because they've got their own DNA. Well, this happens to be one of the few places where you can actually see it. That kinetoplast is a lump of DNA inside a big mitochondrion that fills up the front end of the cell right next to where the flagellum attaches. And there you've got the kinetoplast right there. It's a funny thing. It's made up of these interlocked circular pieces of mitochondrial DNA instead of just one loop of DNA. These guys have mitochondrial DNA that's made up of dozens of so-called maxi circles and thousands of mini circles, all linked together, like on that day when you had a box of 100 key rings and you got really, really bored. I don't know if you ever did that, but imagine what a really bored person with a box of key rings would do. That's kind of what the kinetoplast is, all these interlocking circular pieces of mitochondrial DNA. Kinetoplastids can actually take many forms. This one's called a tripomastigote. Trypanosoma is a nice example of a tripomastigote, and you can see how the flagellum is attached to the cell body by that fin. In others, the flagellum isn't attached by that fin and just sticks out freely. That's a promastigote. Leishmania goes through a stage in the human body where it doesn't have a flagellum at all. That's an amastigote. In the fly, Leishmania does have a flagellum. There's some where the flagellum is attached deep inside the cell in a depression called the flagellar pocket. Never mind. probably not critical for what we have to do here, but trypanosome or kinetoplastids can take on different forms. Even the same species can take on different forms depending on what part of the life cycle we're talking about. My favorite excavates, that's not a sentence you hear very often, are the ones that I ended up studying back when I too was a humble undergraduate. My senior year, I did an honors thesis because you could do that back then. And I happened to work with a expert in protists, specialist in protists of soil, for example. You don't think of this a lot, but there's actually a pretty good-sized assemblage of protists that live in damp soils. And some of the ones that you find, and I know this because I isolated them from the soil around the roots of some weeds outside my apartment in New Orleans. In that soil, you find excavates in a group called the Heterolobosea. And these are really neat because they can switch back and forth between amoeba forms, just little crawling amoebas, and they'll feed on bacteria as long as they can. And when the food starts running out, they kind of firm up and stop swimming around, crawling like amoebas, and they firm up their membrane, and they sprout some flagella, and they start swimming. And in the lab, you could actually make them do this. You could grow the amoebas on an agar plate, and all you had to do to make them transform was flood the agar plate with some distilled water, and within a couple of hours, boing, the amoebas would turn into flagellates and start swimming around. And when they settled down, and I actually got to see this once they find a good place, I suppose. I don't know why they do it, but when they're done with being flagellates, they just start absorbing their flagella and loosening up the cell membrane, and they transform right back into amoebas. And they can do this reversibly as many times as you please. So kind of a really neat trick there. They can also form resting cysts if conditions are bad. The amoeba form will just secrete a wall around itself and just sit there and wait for possibly up to months. I'm not sure what the record is, but they'll just sit there and chill until conditions improve. Now, I worked on a species called Niglaria gruberi, which was not particularly dangerous. But there's a species that lives in warm water that happens to be about body temperature. You would not find these in cold streams or ponds, but you can find them in ponds that get a lot of sunlight, for example, that may have a water temperature that's closer to the human body temperature. If you get those in your body, they can do things like this. Niglaria fowleri is the species that tolerates warmer temperatures. And if you get this up your nose, the amoebas will travel up your olfactory nerve, the nerve of the sense of smell, up into the brain, and start doing this to your brain, causing these terrible damage to your brain. Fortunately, this is extremely rare. Between 1998 and 2007, there were only 33 US cases. You really have to be unlucky to get this. Most of the cases come from swimming in contaminated water. Chlorine gets rid of these, and they're not going to grow in comparatively cool water. But if you're swimming in a warm swimming hole and you get water up your nose, you're at greater risk. Although even then, you kind of have to be unlucky to have this happen. The problem with it is that, well, first you start losing your sense of smell, and then you start getting flu-like symptoms, and then you might start getting a stiff neck and a headache, and then you start showing neuro symptoms. By the time the doctors realize that this isn't just the flu, the damage has actually progressed pretty far. And amoebic meningoencephalitis like this is almost always fatal. There aren't very many good treatments for it, in part because it's so rare that many doctors never even see it. And in part that by the time it's obvious what it is, it may be too late. You have to diagnose this by doing a spinal tap and looking for amoebas in the cerebrospinal fluid. And this is actually a matter of local interest because in July 2013, a 12-year-old girl named Callie Hardig actually got this at Willow Springs Water Park, which is closed now. This is part of the reason why. They didn't treat their water. They let their water get warm. There were actually 2 people that caught this, and one of them died. But Callie Hardig was treated at Arkansas Children's Hospital. It was big news because they had to get emergency approval for a drug that had been developed in Germany, but that isn't yet approved in this country. And they had to express fly some of that drug from Germany to here. And fortunately, Callie Hardig became only the 3rd person in 50 years to survive amoebic meningoencephalitis. She did have some brain damage when she came out of the coma she was in. They put her in an induced coma to try to slow it down. And last I heard, there was a follow-up news story on her a few years ago. And last I heard, she had pretty much recovered, didn't have any lasting neuro damage, and was starting to look into colleges. Yeah, she was extremely lucky, and Arkansas Children's Hospital is pretty good because she was only the 3rd survivor of this in the past 50 years. So there, little local connection. If you are concerned about this, nose clips, and you won't have a thing to worry about. Oh, there were another couple of cases of this that were caused by people using contaminated water in a neti pot. This is not the kind of thing I enjoy doing, but apparently a neti pot is this pot with a long spout, and some people pour warm saline into a neti pot and use it to irrigate their sinuses. I do not personally do that. If you ever do this, make sure you're using sterile saline to irrigate your sinuses. If you use a neti pot, dirty saline can put these amoebas up your nose. See, never let it be said I don't teach anything useful. Right, that's my piece about excavates. I won't say very much about the clade Archaeplastida, which is also sometimes called plantae. Although you'll see plantae sometimes used for just the land plants, you know, mosses and trees and flowers and all that. But some people seem to use plantae for the group that includes the land plants, the green algae, the red algae, and some minor groups. I usually call it Archaeplastida. I don't care what you call it. We're not going to have very much to say about them. The chloroplasts in this group are surrounded by only 2 membranes, and other algae have chloroplasts surrounded by 3 or 4 membranes. I mentioned that dinoflagellates have chloroplasts that may be surrounded by 4. Apicomplexa have 4 membranes around their apicoplasts, which look like chloroplasts that are no longer used for their original function. And what we think is that the ancestor of this group, the Archaeoplastida, is the only major branch that formed a primary symbiosis with blue-green bacteria. This is the branch where an ancestor said, hey, what happens if I engulf a blue-green bacterium but don't digest it right away and just see what it does? The ancestors of most other algal groups either engulfed an archaeoplastid and stole the chloroplasts, or in some cases they engulfed an archaeoplastid and retained the entire cell. There's several groups of algae. There's one called the cryptomonads, in which the chloroplast is actually surrounded by 4 membranes, and it's got a trace of the host nucleus. Well, it's got a nucleus of its own, and it's got a trace of a eukaryote nucleus and a chloroplast DNA, meaning that it's basically descended from a eukaryote. I'm not explaining this very well, but this is Russian dolls again. The host engulfed a eukaryote that had a chloroplast. And traces of all 3, the chloroplast, the original host, and the new host are still there. That's called secondary symbiosis, when you borrow your chloroplasts from another organism. This is a small group of protists called glaucophytes, and they're important because their chloroplasts still have bacteria-type cell walls. The chloroplasts of, plants and green algae and red algae no longer have their own cell walls, but these still do. There's a red alga. It's red because it's got extra pigments that are called phycobiloproteins that help it absorb light of different wavelengths. They absorb blue light efficiently. The other main plantae lineage is sometimes called the viridiplantae. That's the land plants and green algae, including this very handsome green single-celled alga of a type called a desmid. And it's possible that you could see these if you look at enough pond water. They're grass green, and they're always very symmetrical like this. And anyway, green plants and green algae use 2 types of chlorophyll, A and B. And that's all I'm going to say about that. Tell you what, this is actually a pretty good place for a break if you want it. Tell you what, I've got 9:54. Take 10 minutes to do whatever and come back at 10:05 and we'll reconvene. Okie dokie. Let me check the chat real quick. Don't seem to be any new questions, so I'm going to go ahead and stop recording right now.